Hi, everyone. My name is Mary Wuerth, and welcome to Back to School Night at Tamalpais High School 2021. And welcome to Biomedical Sciences at Tamalpais High School. My name is Mary. I have my bachelor's degree in biochemistry from UCLA and a master's degree in biological sciences from Clemson University. And before we go on, I want to make sure to thank the Tam High Foundation for their support. Tam High Foundation makes a lot of the great labs we do in the science department possible. In fact, I just ordered, I think, $2,000 worth of stuff for labs for this class last week, and we couldn't afford to do that without the support of the Tam High Foundation, so thank you. A little bit about me. I just finished my 32nd year teaching. 28 of those have been at TAM. 6 of those I was also adjunct picking up an occasional night class at College of Marin in the Life Science Department. In 2003, I was invited to the White House and received the Presidential Award for Excellence in Science Teaching. I currently serve as a biointeractive ambassador for HHMI. Before the pandemic, I would go around the country teaching workshops to science teachers on how to use the latest research and some of the biointeractive activities in their classroom. Right now, we're doing it all remote, of course. In fact, this Saturday, I'm teaching an online workshop for teachers on how to teach about CRISPR with their students. And yes, we will be doing CRISPR at the end of the year in this class. For those of you who don't know, I am married to Mr. Baugh, who teaches AP English Lit at TAM. We didn't meet here. We met when we were both teaching in Los Angeles. Mr. Baugh grew up here and convinced me in 1993 to move from Southern California to Marin, and we've never looked back. We have 2 daughters who are 14 and 16 years old who go to Terra Linda High School. Okay. Now, as a parent, one of the first things I want to know is, how will I know what's going on in class? If you go to my website, wuerthbiomed.weebly.com, you'll find daily agendas and links to all the documents that we're using in class. And that website is also listed on my Canvas homepage. Another thing I want to know as a parent is, how is my student doing in your class? Well, grades will be posted in Synergy. I try to update those every Friday or Saturday. Synergy is a brand new system to the TAM district this year, so I appreciate your patience as we all learn how to navigate it. It's been a tiny bit buggy, but they seem to be getting most of those bugs taken care of. Okay. Another thing I want to know as a parent is, what should my student be doing when they get home from school? Now, the homework level in biomedical sciences is less than it is in the other class I teach, which is advanced placement biology. But there still are times when studying should occur. When they want to review concepts learned in class, when we have a quiz or exam coming up, and those exam dates and quiz dates will be posted on my website. Also, every Monday, we have Monday seminar where we will take turns presenting the scientific paper to the class and discussing it. The paper is posted on the website on Thursday. All students are expected to read the paper and to come to class on Monday with at least 5 questions to share during the class discussion of the paper. All students need to keep a spiral-bound laboratory notebook. We spent some time setting that up in class the first week of school, and I had already provided spiral notebooks for anyone who didn't have them. So your student should already have that all set up. Also, each semester, in lieu of a final exam, your student will be doing a job shadow, which, due to COVID, is going to be virtual this year, probably a phone interview. Fall semester, they need to find someone who's working in the research field. Spring semester, they'll repeat the process and they will find someone who's working in a clinical setting. I've already provided them with a planner with different benchmarks, different due days, breaking it down for them, and some sentence starters for when they make their initial phone call. And they will be getting some time in class to make those initial phone calls to find someone that they can phone interview for their job shadow project. Now, we just finished our first unit, our little mini unit on public health, and we focused on the biology of the virus that causes COVID. And we had a Monday seminar discussion of a paper comparing patient outcomes for people who were hospitalized with the side effects of COVID who got convalescent plasma. Our unit that we just started this week is on genetics. The theme, organizing phenomena we're going to use is how do we use the technology we have in forensics, and how do we use the technology we have to help people who have been wrongly accused? And we talk a little bit about the Innocence Project. In the lab skills, your students are going to learn, they're going to learn how to make and run electrophoresis gel to separate DNA fragments, and how to do a forensic DNA test. Next week, we're just going to do a simple introductory lab where they're going to cut some DNA up with restriction enzymes, run it on the gel. But later on in this unit, they're going to learn how to do PCR because we have a thermal cycler. Our next unit will be on immunology. How does our immune system work? Why do some people go into anaphylactic shock when exposed to certain allergens? What are autoimmune diseases? How do we test for the presence of antibodies? Students are going to learn lab skills on how to detect antibodies qualitatively and quantitatively. Unit number 3 is going to be on bacteria. We'll talk about gram-negative bacteria, antibiotic-resistant bacteria. Why are they a problem? When is it appropriate to use antibiotics and when is it not appropriate? Your student will learn how to culture bacteria, stain and differentiate between gram-positive, gram-negative bacteria, how to test for the effectiveness of different antibiotics, and we're going to make a transgenic bacteria. We're going to use Biorad's pGlow kit to introduce a new gene into some bacteria, so that bacteria, a GFP gene, so that the bacteria will glow in under UV light. It's kind of a fun, cool lab. Our third unit will focus on viruses. We'll talk about some of the history of HIV. How do you test for HIV? What are some of the treatments for HIV? What's a virus? Are viruses living or not? We'll do ELISA tests and Western blot tests in Unit 4. And then our final unit is going to be on cancer. What is cancer? Why is it so scary? How can we do, what genes are associated with increased cancer risks? And how do they test for them? And we'll talk a little bit about genetic counseling, which I think is also an interesting career for students to be thinking about. We'll look at biopsied cells under a microscope, learn how to distinguish between normal and cancer cells, and how to run a gel to detect the presence or absence of genes related to cancer. Okay. Thanks so much for coming this evening, albeit virtually, and for listening. If you have any questions, please don't hesitate to contact me at mwirth@tamdistrict.org. And again, thank you to the Tam High Foundation for making this all possible. And thank you and have a great evening.